Hello, my name is Sonya Dyhrman. I'm a professor at Columbia University in the Department of Earth and Environmental Sciences at the Lamont Doherty Earth Observatory. And thank you for joining me today for this presentation, which I've entitled Defining the Niche, Tracking Coccolithophore Transcripts in the North Pacific. In my lab group, we're broadly interested in how phytoplankton interact with their environment and how they drive ecosystem structure and function in the broader ocean. And to do that, we often use molecular level tools. Before I begin, I want to especially acknowledge my former graduate student, Harriet Alexander, for her assistance with the graphics. So as I said, we study phytoplankton in my group, and their complex community dynamics really that drive phytoplankton physiological ecology in the ocean. For example, light availability, predation and loss terms, cell-cell interactions and exchanges, as well as what we would call bottom-up control or nutrient availability. And today what I'm going to be talking about is nutrients and light and how they drive the dynamics of the phytoplankton E. huxleyi. Now, we care about these phytoplankton dynamics because they play an important role in ocean ecosystem function and biogeochemistry. For example, as they're photosynthetic organisms, they drive carbon fixation and the flow of carbon into the food web. Now, some of that carbon in the ocean food web is respired back out to the atmosphere, but some of it is exported to the deep sea. And so these phytoplankton play a critical role in the ocean carbon cycle. The work that I'm going to be talking about today is all focused on the North Pacific Subtropical Gyre, or NPSG for short. It's arguably the largest biome on the planet, and in these low nutrient waters, even though the phytoplankton are perhaps not as dense as shown in this chlorophyll image here as in other environments, because it is such a large swath of ocean, it is critically important to study. Another reason why we focus a lot of our work here is that the U.S. runs a time series site at a place called Station Aloha, which is north of the Hawaiian Islands, and runs regular monthly time series analyses at this location, which allows us to compare our work to the historical record and really frame it in this broader observational understanding. Now, because of all the work that's gone on in this environment, we know that it's dominated by small picoeukaryotic phytoplankton and picocyanobacteria, and that they drive a very active microbial loop. And that the groups that I'm going to be talking about today, and specifically E. hux shown in this cartoon here, which are larger, greater than 5 micron microeukaryotes, are at much lower abundance than their small counterparts. However, under certain conditions, these organisms can play a critical role in this ecosystem by driving increased carbon export. That's because they can either proliferate or bloom with additions of resources like nutrients, and because both of these forms in this example biomineralize, silica with the diatoms and calcification with the haptophytes, they're large and they're heavy, and so again they can drive carbon export processes and thus play a disproportionately important role in the carbon cycle dynamics of this region. Now, the species that I'm going to be talking about today is E. emiliana huxleyi, or E. hux for short. It's broadly distributed and globally significant. It can form dense blooms, such as the ones shown from outer space here, and it's why the White Cliffs of Dover, shown here, are white. That's because it's a calcifier or a coccolithophore, and as a result, it plays a critical role in the global carbon cycle and is strongly linked to climate-driven ocean acidification and for future ocean scenarios. It's also a source of paleoproxies for climate reconstructions, is known to form dense blooms, the drivers of which are largely unknown, and there are whole genome sequences from multiple strains of E. huxleyi which identified a pangenome. This not only gives us a great sequence resource to work with to do our analyses, but this idea of a pangenome is important, and I will come back to that at the end of the talk. So today I'm going to be talking about what factors drive community structure and function in the NPSG with a specific focus on the E-hux, or defining the niche for that keystone taxa, focusing first on light and then on nutrients. Some of the questions that we're focused on for later are there diel or day-night driven transcriptional patterns in E. huxleyi, where it partitions what it's doing over the course of the day, and are there group-specific responses to day-night cycles that might allow E. hux to distinguish its niche from that of its phytoplankton neighbor? To do this work on light, we were part of a large synthesis experiment supported by the Simons Foundation through the Simons Collaboration on Ocean Processes and Ecology. This diel process study involved a research cruise off the coast of the Hawaiian Islands, shown here. And in the study areas in the box here, we deployed a drifter into this eddy and tracked it so that we could sample the same water mass over time. Here, the dots indicate sample times and locations, and each of the dark dots means we sampled at night, and the yellow dots mean we sampled during the day. Our general approach looked like this. We sampled the greater than 5 micron phytoplankton community, isolated RNA, did a polyase selection to pull out the eukaryotic signals, and sequenced it to a targeted depth of roughly 60 million 100 base pair paired end reads. We then took those reads and mapped them stringently to a custom database of transcriptome sequences from many hundreds of different species of phytoplankton that are present in the MMETSP, or the Marine Microbial Eukaryote Transcriptome Sequencing Project. It's a mouthful, which was funded by the Gordon and Betty Moore Foundation. So what does our sampling actually look like? It looks something like this. It's sort of controlled chaos. You can see we have all of these different pumps going, we have different lines, we have these big, large volumes of water because the populations we're working on are quite dilute. And here you can see we run multiple filters all at the same time. And for this diel study, we collected samples every 4 hours for 4 days, twice. Not only during the daytime, as shown here, but also, if you recall, we were sampling at night, which meant working and filtering in the laboratory under red light to try to preserve the diel signal we were looking for. So I need to especially acknowledge my technician, Sheehan Haley, shown here, and the other members of my lab group that were on this cruise with me, because it was quite an effort. So our first question, in fact, what kept me up while I was sampling in the middle of the night, was, are we even going to be able to detect E. huxleyi in these dilute populations? And thankfully, the answer was yes. So here in this plot, we have the breakdown, the taxonomic breakdown of distribution of transcripts over time based on different phytoplankton groups. And the haptophytes are shown in red, and E. huxleyi is shown in the very dark red bar up here. So you can see it's not a large component of the transcriptional signal, but it is present. Just to orient you for this and future plots, the gray bar means we sampled at nighttime. The white bar means we sampled during the day. And so over time, you can see a very consistent presence of these taxa across time. We detected a diverse community of phytoplankton The signal was dominated by dinoflagellates in this yellowy color because they have large transcriptomes. And in fact, the highest haptophyte signal, so the dominant signal within the haptophytes, was from E. huxleyi. I want to mention that this work that I'm talking about is published in Environmental Microbiology with lead author Maria Hernandez Limon. Maria was a bridge to PhD scholar in my laboratory and is now working on her PhD at the University of Chicago. So we could detect EHUX, and the next question, of course, is could we mine that transcriptional signal to look for diel patterns, and could we even detect them? So again, just to remind you that this is a challenging task. We're sequencing the whole community, and then we're trying to find diel signals that are going up and down for functional genes within a specific E. huxleyi species. So again, I was a little nervous that we wouldn't be able to detect much in terms of a signal, and thankfully, as you might have guessed, we were okay. We detected an amazingly beautiful transcriptional signal where you can see all of these transcripts going up and down in E. huxleyi over the course of a day. So I know this heat map is a little busy, but just again to orient you, we have our night signal and our day signal across the top, and each of these lines is a transcript going from minimum to maximum over the course of day-night cycles. And you can see all this beautiful phase transcription of E. huxleyi as it's doing its thing over the course of the day. These data are clustered, statistically clustered, and that's what's shown in these colored bars here, with the average pattern, for example, of this blue cluster shown here on the right. So you can look at it in more detail if you want in the video. But you can see, again, we have a large number of transcripts that are under diel patterns in E. huxleyi for this data set. And we can go in and ask, well, what functions are changing over the course of the day? And I just wanted to briefly show you a couple of examples here for carbon pathways and some fatty acid pathways. So here you're looking at normalized expression over time with day and night signals, and the line is the average signal of all transcripts in the pathway. The envelope or shading around that line is the standard error, and the numbers in parentheses just indicate the total number of transcripts in that pathway that were either significant or not significant. And you can see that during the nighttime, E. huxleyi starts to ramp up its transcription of photosynthetic and carbon fixation related transcripts so that they are turned on in the morning when the lights come on. And the TCA cycle shown here in the brown peaks later in the late afternoon, and you can just again envision the carbon being fixed and then moving through metabolism over the course of the day for these populations. We see phase transcriptional patterns for pathways in fatty acid metabolism as well. For example, biosynthesis, followed by elongation and degradation later in the day. I honestly never get tired of looking at these pathways because it's still kind of amazing to me that we can pick out and really try to understand what this one species of phytoplankton is doing over the course of the day in the environment. And just to summarize what we found here for E. hux, roughly 60% of context is clustered into groupings or modules with statistically significant diel periodicity. So a large fraction of our signal had a diel pattern. There are also patterns in metabolism that suggested the orchestrated cycle of building and then consuming energy stores. You saw those patterns with carbon fixation and the TCA cycle as the cells go from the day into the night. There were diel patterns in contigs or transcripts associated with the cell cycle that were consistent with the timing of cell division observed in culture. There were diel expression of urea and ammonia transporters that suggest a potential phasing of nitrogen transport processes throughout the day and night for this species, and in aggregate, putative calcification contigs did not show a significant diel signal. The calcification pathways are still poorly understood for this species. Now, we were able to pick out and understand what E. hux was doing. One question is, are all phytoplankton doing the same thing in this environment, or are E. hux or the haptophyte lineage doing something unique from other lineages? And to address that question, we have been looking collaboratively with work led by my former postdoc, Matt Harkey, who's now at the Gloucester Marine Genomics Institute, in collaboration with other Simons Foundation investigators and their groups, including Joshua White, who's leading the synthesis effort, and graduate students Daniel Moratori and Angie Boyson. And what you see here is the transcripts from our data set, this time integrated across different KEGG pathways across all taxa. So this is the community signal in an NMDS projection. And here each dot represents a transcript, and you can see it really looks like a clock. So each of these dots represent the peak time for that transcript, and you can see as you go through the course of the day, the whole community is changing its diel signatures, and genes are coming on and genes are going off as you move throughout the course of the day. Now, one question is, this community signal, would it be the same if we were to only look at the haptophytes, let's say, or the diatoms? For example, do different groups do different things even when they share a metabolism? And the short answer to that is yes, they do different things. So this is the same kind of projection where each of the dots represents a transcript, but this time, instead of color coding it by time of day, we've just pulled out all of the signals for the diatoms and for the haptophytes. And you can see right away that different groups have different patterns. The E. huxleyi signal is embedded in this, and you can see that this haptophyte pattern around that circle does not look the same as the diatom signal. Now we can delve in even further and ask about different haptophytes and different diatoms and whether they're doing the same thing. And in ongoing work, this really supports the idea that diel responses are congruent between genera and lineage specific. So let me walk you through these data a little bit more closely. All of these left-hand panels are the photosynthesis, carbon fixation, and TCA cycle that you've seen before. And because these organisms are photosynthetic, you can see that these patterns are highly similar regardless of whether you're a haptophyte or a diatom, and regardless of which genus of haptophyte or diatom you were looking at. But other metabolic pathways, for example, transcripts involved in cell cycle, would show a strong, similar diel signal for both Emiliania and this other haptophyte, Chrysochromulina, don't have that same diel signal for Rhizosolenia and Cylindrotheca in the diatoms. So once you get away from photosynthesis, there are differences between lineages, but those differences really don't seem to occur between different genera, at least at this juncture in the analysis. So you can envision that there may be temporal niche partitioning between haptophytes like E. hux and its diatom neighbors within the community, and that this data supports that hypothesis. So, to summarize, E-Huxley transcription is tightly phased to light-dark cycles. We had a beautiful cycling of many transcripts in E-Huxley that really defines its niche with regards to the day and night cycles, and that lineage-specific signals on shared pathways may indicate temporal niche partitioning between lineages, such as the data that we looked at between the diatoms and the haptophytes. Now, next, I'm going to be talking about nutrient dynamics, which nutrients are important to E. huxleyi in this environment, what is its physiological response to a shift in nutrient environment, and how do nutrients influence diversity. For that work, we also did sampling in the NPSG and ran experiments near Station Aloha, where we did large-scale incubation experiments designed to shift the nutrient environment for these populations. These were semi-factorial resource addition experiments, where we have an in situ sample, a control sample with no amendment, and then we had 3 treatments where we increased the N to P ratio, either by adding deep, resource-rich seawater, adding nitrate, or adding nitrate and other resources, really to drive the physiology of that treatment to a low phosphorus condition. As a counterpoint, we also had treatments where we decreased the N to P ratio. For example, this treatment where we added phosphate and other resources, again to drive the physiology now more towards a low nitrogen scenario, and we added phosphate. Our sampling for these experiments is very similar to that which I showed you before, but now we're sampling incubation treatments after a 6-day incubation. So first we'll look at the general transcriptional patterns that we saw in treatments versus the control for E. huxleyi. So this is log fold change versus the log average abundance, where each dot represents an E. huxleyi transcript in this environment. And the yellow dots that you see here are transcripts that were significantly differentially expressed across the 2 replicated experiments that we did in this system. I should mention that this work is published in Environmental Microbiology with lead author Harriet Alexander, who, as I said earlier, is a former PhD student and now a scientist at Hui. Now, first of all, what should be striking is that we really only saw a significant differential transcriptional response in treatments where we added nitrate and increased the N to P ratio. You can see all of these transcripts going up and all of these transcripts going down, and next to no differentially expressed signal in the other treatments where we decreased the N to P ratio. So this suggests a strong end control of transcription and metabolic response in this system. Now you might look at this and ask the question, are these the same genes or transcripts that are changing in each of these 3 treatments? And the answer to that is largely yes. So again, this is a very conserved response where the majority of the transcripts that are changing across each of these different conditions are the same. So again, very concerted and consistent response to nitrogen addition. Just to summarize what some of these transcripts are, I will briefly highlight those data here. So here's a little cell schematic where we have the control and then the treatments with added nitrate on the right. The signal in orange color are gene targets related to P metabolism that are generally regulated by phosphorus availability. Similarly, in the teal color, we have N targets that are genes that are regulated by N physiology. And in the dark blue are genes related to calcification. So what you can see is that there's a general decrease in N metabolism transcripts. And this makes sense. If you have a population existing in a low nitrogen environment, working very hard to acquire nitrogen and really pulling out all the transcriptional stops, so to speak, and you add nitrogen, that nitrogen will mean the cells don't have to work so hard to acquire nitrogen, and we typically see a downregulation of the N metabolism transcripts as a result. In contrast, we see here a general increase in P metabolism transcripts. And this suggests that phosphorus is secondarily limiting in this scenario, because when we add nitrate and relieve that stress, we're driving the population to P stress, and that's why you see the increase in these P stress markers as the cells have to work harder and harder to acquire phosphorus. And what was exciting for us is that we also saw a general increase in putative calcification genes, which suggests that these cells are either increasing calcification or shifting from non-calcifying to calcifying life stages. So to summarize, nitrogen addition elicits a really robust transcriptional response in E. huxleyi, where phosphorus might be secondarily limiting. Nitrogen appears to be a major controlling factor for these populations. And the transcriptional data suggested there might be a shift between either calcified and non-calcified forms or an increase in calcification with nitrogen addition. To finish here, I wanted to end with this final question, and that is, how do nutrients influence EHUX diversity? And for that, I want to go back to the idea of the pangenome and really work to define the role of the pangenome in E. huxleyi physiological ecology in these populations. So the idea of a pangenome means that you have a core set of genes that really define E. huxleyi. All E. huxleyi strains carry these genes in their core. But each E. huxleyi strain also carries a set of variable genes, which may be shared by other strains but are not found in all strains. So you might envision a scenario that we would hypothesize that might look something like this. If you looked out into the environment, there might be a dominant strain with a pangenome or a variable set of genes that really helped it thrive in a given environment, even though other strains might be present. And this idea of the variable gene content helping and really driving how these different strains are distributed in a given environment is something that we try to look at with our transcript data in the incubations I just told you about. And to do that, we collected available transcriptomes from E. huxleyi strains that were isolated across very different environments all over the global ocean. We clustered those transcripts into orthologous groups in order to identify core transcripts, variable transcripts, and strain-specific transcripts, or I might call them OGs for short for orthologous groups. So what the data look like are something like this, where we have a core set of genes and a variable set of genes, although we're defining this at the transcript level. And the distribution of those genes looks like this, where you have the number of orthologous groups here on the y-axis, and you can see that there are a number that are core here, and then there are a number that are variable, meaning they're only found in one or more strains, not in all strains. And then the colors here on the right are strain-specific OGs. So these are transcripts that are only found in the given strain and can be used to designate a strain-specific signal. So what we did is take this data and use it as a reference database and take all of our sequence reads from EHOCs that we collected in those incubations in the North Pacific and map them to this data set to examine shifts in expression over time in this environment and in the incubations. First, we wanted to look at the core versus the variable responses, and then I'll show you the strain-specific dynamics. For the core versus variable responses, when you think about it, we've been arguing that that variable gene set is really what drives a strain's ability to thrive in a given environment. And you could postulate that if you change the environment by adding nutrients, for example, that you would see a shift in the expression of the variable gene set. So it's those data that we'll look at next. And to make a long story short, the variable portion of the pangenome for is selected for in our nutrient addition experiment. So here we have the percent of reads mapping to the variable gene set. Our in situ signal before we added the nutrients was about 20%. And in green, all those treatments that got a nitrate addition, you can see that there's a significant increase following nitrate addition in the expression of that variable gene set. So there is selection for the expression and modulating the expression of the variable gene set in altered environmental conditions, which really speaks to the fact that these variable genes might be important in how these strains thrive in a given environment. We also looked at the unique or strain-specific gene sets. They co-occur in the environment, as you can see in this first graphic. So these are samples in situ over time during our study. And these are the percent of reads mapped to E. huxleyi. And you're just going to focus on these strain-specific signals, which are color-coded. So you can see the majority of the signal comes from this yellow strain, which has the highest signal in this environment, and is in contrast to this blue-colored strain, which has a very low signal in this environment. So the question is, if we add resources, do any of these patterns change? And again, the answer is yes. So here we have the same percent of reads mapped to E. huxleyi. And if you just focus on the yellow and the blue here, you can see the yellow is highest in situ in the control and in treatments that didn't receive nitrate. And that signal is dramatically reduced in the samples that received nitrate. That's opposite from the blue strain, which in the environment was very low, but increased dramatically when we added nitrate. So what this suggests is that strains may sort themselves in the environment based on the content of their variable gene set as a way of thriving under different conditions. And when we manipulate those conditions, you get, in essence, a different winning strain. This was really tantalizing work done at the transcriptome level, and what we really want to do is look at this at the DNA or gene level. And to do that, we're actually in the process of sequencing 15 new genomes from E. huxleyi isolates that are in culture collections that have been isolated from throughout the world oceans, shown here in the red dots. And this increase in the availability of sequenced genomes will really allow us to better define the pangenome and what makes up the variable gene set. And this is part of new research that's being led by Harriet Alexander, where again we're defining the role of the pangenome in E. huxleyi physiology, ecology, and biogeography. First, by defining the pangenome with the genome sequencing, then looking at whether or not we have that ecotype or strain sorting in the environment, and then using culture controls to characterize the response to changing environments and to see whether or not we can isolate it to the variable gene set. So to summarize, we've been defining the niche for E. huxleyi and other species in the NPSG, and I hope I'm leaving you with the idea today that it's an exciting time to leverage these molecular tools to track the physiological ecology of these phytoplankton communities and their role in ocean ecosystem structure, function, and biogeochemistry. I'd like to conclude with acknowledgments both of my laboratory, my collaborators, and funding, in particular Sheehan Haley, Matt Harkey, Harriet Alexander, and Maria Hernandez Limon for their contributions to the data that I've presented in this talk. Thank you for your time.